Hi, I'm John Zehr, and I'm from the University of California, Santa Cruz, Department of Ocean Sciences. And I'm going to talk to you about nitrogen fixation, specifically about symbiotic system, uh, nitrogen fixing symbiotic system in the open ocean. So first, a little bit of background about nitrogen fixation, some important things to know. Um, it, Nitrogen fixation is important in terrestrial ecosystems, of course, known in agricultural terrestrial ecosystems for centuries. There are diverse bacteria and archaea that can fix nitrogen, and some of them are symbiotic. This is true in terrestrial environment as we'll see in the marine environment. And the only known nitrogen fixation that occurs in eukaryotes occurs in symbiosis with bacteria or archaea. And in the terrestrial environment, it's really common that multicellular plants form symbioses with nitrogen-fixing organisms, such as the legumes shown in the lower panel here that have nitrogen-fixing bacteria associated with their roots. But there's many and diverse different types of symbiosis in terrestrial systems. So nitrogen fixation in the marine world was really assumed not to be of particular interest in the early years of marine biogeochemistry and marine science. And the process was known to occur, and it was known that there were some nitrogen-fixing bacteria, for example, that lived in marine water. But it was assumed that it wasn't all that important because it was assumed that nitrogen wouldn't limit the growth of organisms or biological productivity because nitrogen fixation would just make up any deficits, and that the real interest would be in the primary limiting nutrients, which was largely to be phosphorus. So these are some early Redfield papers that make this case that really phosphorus is an important nutrient. Nitrogen fixation is not that important. It's assumed to occur, but it's assumed to occur at low rates just to sort of top up any deficit that there might be in nitrogen needs. And then in the 90s, Ed Carpenter and Doug Capone, following Dick Dugdale's discovery that trichodesmium fixed nitrogen in the 1960s, showed that there were actually substantial rates and even blooms of trichodesmium, largely in tropical and some subtropical waters. And then near the end of the 90s, as biogeochemists started looking at the balances, they came up with this imbalance, which I think Bess Ward talked about, suggesting that there was a missing source of nitrogen fixation to balance the known losses due to denitrification. And so it was thought that, well, some of these might be missing either known nitrogen fixation habitats or organisms or some source of nitrogen that we didn't know about. So what we know now is that nitrogen fixation is important to surface oceans. There's natural nitrogen fixation in the sea that roughly is equivalent to the natural nitrogen fixation on land. Agricultural nitrogen fixation adds nitrogen to terrestrial environments. But in the ocean environment, particularly in oligotrophic oceans, it's now known that nitrogen fixation supports a large fraction of net community production. So a little bit about the process, because nitrogen fixation is really an interesting reaction, and the organisms that can do it are really interesting. This is the fundamental biological chemical reaction for nitrogen fixation. Nitrogen gas N2 reacts with ATP, hydrogen ion, and electrons to form ammonia, and a hydrogen molecule is released, as I will mention again. And the ATP, of course, is the energy is used and forms ADP and inorganic phosphorus. So the important thing is that this reaction requires a lot of stuff, biochemical stuff. It needs ATP, so that's an energy drain for the cell, and it needs reductant in the forms of electrons, or NADH, wherever the source is. So it's energetically for the cell a rather expensive process. And it's carried out by a really cool but complex enzyme. This enzyme is actually composed of two separate multi-subunits proteins. What's called the iron protein, which is the smaller one composed of two identical subunits, and the molybdenum iron protein, which contains two different types of subunits. Both of them have metal clusters. The iron protein has an iron sulfur cluster, but as you can see, the molybdenum iron protein has got these other metal clusters, for example, called the P cluster, that contain iron and are very important in the electron transfer. So this is the enzyme that actually catalyzes the reduction of nitrogen gas to form ammonium. It reduces hydrogen ion to H2 at the same time, and it can't really be prevented, so it's an energy loss. Some of that can be recovered by some nitrogen-fixing organisms, by uptake hydrogenases. But the important thing for our discussion here today is that this enzyme, because of the iron sulfur centers and the need for electron transfer, is rapidly inactivated by oxygen within seconds. And so this presents an interesting paradox for some microorganisms, such as cyanobacteria, that are photosynthetic and are evolving oxygen, because they are evolving oxygen, but yet the nitrogen fixation that they catalyze, not all cyanobacteria fix nitrogen, but some of them do, is so sensitive to oxygen. How can this be? So it's a problem for organisms to resolve this, and for biochemists to figure out how this works. In cyanobacteria that are photosynthetic and evolve oxygen, we know that there's two main strategies. Either they separate the two activities of photosynthesis, evolving oxygen and nitrogen fixation, by day versus night, fixing nitrogen during the night when they're not evolving oxygen, or they make specialized cells called heterocysts, as shown in the top panel here, where nitrogen fixing conditions are maintained without oxygen. Oxygen is not evolved, and they fix nitrogen, or as photosynthesis occurs in what's called the vegetative cells. So most organisms fall along one of these patterns that can be explained either by temporally separating nitrogen fixation from photosynthesis, such as here in a unicellular cyanobacterium called Cyanophyceae, or in heterocyst forming cyanobacteria that are essentially multicellular and make specialized cells for nitrogen fixation. So since there's multiple proteins, there's also multiple genes, but it's even more complex than that. So there are genes for the molybdenum iron protein, there are genes for the iron protein, but there's all this other alphabet soup of genes that are also required in order to have active nitrogen fixation. So these are involved as scaffolds to put together this complex enzyme, or transporters, or involved in making the cofactors, the iron sulfur centers, and so on. It's a very complex set of genes, and this is described in a chapter in a new book that should be coming out this year. There's many different papers you can read about this if you're interested. There's also different forms of nitrogenase, one that is dependent on the molybdenum metal, and other nitrogenases that replace the molybdenum with vanadium or even iron. And so then they have iron plus the iron sulfur centers that are in the molybdenum and vanadium. And so that's another set of genes. They're homologous to or similar to the molybdenum genes, but it's yet another set of genes. And then there's other sets of genes that are symbiosis genes in those terrestrial organisms that form nodules, for example, with plants. So there's a lot of different genes that are necessary for nitrogen fixation. And the organisms that can fix nitrogen are really diverse. They're scattered throughout all the known phylogenetic parts of the tree, except for eukaryotes. And they are diverse in their types of metabolism. So as we just talked about, cyanobacteria are one group, but among the bacteria and archaea, there are also many different genera of nitrogen-fixing organisms that span strict anaerobes that don't have a problem with the oxygen, to microaerophiles and even aerobic heterotrophs that have figured out how to keep the oxygen low enough so that they can perform nitrogen fixation. So the point is the physiology is really diverse. And the interesting thing is this is sort of randomly distributed throughout the phylogenetic tree of bacteria and archaea. But within specific lineages, for example, in Klebsiella, there are species that have the nitrogen fixation genes and those that don't. And so the nitrogen fixation capability is sort of randomly distributed through the tree. And the importance of this is that it makes it difficult to identify a nitrogen fixing organism unless you can show that it has the nitrogen fixing genes, fixation genes, or that it actually fixes nitrogen. You can't just look at the species identity and identify it as a nitrogen fixing organism in general. Okay, so where are we in the ocean? In the ocean, up until the late 1990s, we knew of a number of nitrogen-fixing organisms, and one of those, of course, was Trichodesmium, which forms these large enough aggregates that you can actually see them with the naked eye. They were called sea sawdust by Captain Cook and the sailors in that era. And we also have known from microscopy, even very early microscopy studies, that there are these diatoms that have the heterocyst forming types of cyanobacteria inside them. And so up until the 1990s, we knew that there were these two major groups and a unicellular cyanobacterium had been isolated, but not really shown to be that important in the oceans, or how widely, it wasn't really known how widely distributed. So this is Crocosphaera. And so the day-night patterns of nitrogen fixation, these diatom symbionts would be fixing nitrogen during the day because they have specialized cells. Crocosphaera would be expected to fix nitrogen during the night because it's a single cell and it evolves oxygen during the day when the light's on. And Trichodesmium is very unusual. It also fixes nitrogen during the light. It doesn't have obvious specialized cells, but a number of mechanisms, including some distribution of labor across cells, have been proposed, and I won't talk about that today. So nitrogen-fixing symbioses are very common in the terrestrial environment, and as we'll see, such as the diatoms, there are some known symbioses in the open ocean. I should also say that there are symbiosis in a number of marine systems, and I'm not talking about such as coral reefs, but I'm just talking about planktonic nitrogen fixation here today. So on the left here, you can see that there are groups of nitrogen fixing organisms scattered throughout the phylogenetic tree. So all the species in one of these highlighted areas don't fix nitrogen, but these groups contain nitrogen fixing representatives that are symbiotic with organisms. So Trichodesmium is not symbiotic in the strict sense, but there are symbionts within these groups. So the point is that there are groups scattered throughout phylogeny and terrestrial environment. In the marine planktonic environment, we've known about these diatom symbionts for a long time. These are diatoms, silica frustule, and inside are these chains of cells, fairly short filaments that have the vegetative photosynthetic cells and then the specialized heterocyst. And up until 1998, those were the main players that we knew were fixing nitrogen, and speculation. There was a lot of speculation. Are there other nitrogen fixing organisms? I remember at the beginning of my career, people were wondering whether Synechococcus might fix nitrogen. And so there's a general question of whether or not there are other organisms that can fix nitrogen. And with the development of molecular techniques, specifically the polymerase chain reaction that happened in the 1980s, especially late 1980s, it was possible to develop cultivation independent techniques to look for nitrogen fixing organisms. And now from the introduction, you know how to do that. You need to look for the nitrogen fixing genes, and the polymerase chain reaction provided a way to do that. Since then, there have been development of more molecular biology methods that we'll be talking about, that such as quantitative PCR and metagenomics, that have also allowed us to look for nitrogen fixing organisms. So it's going to try to keep this talk short. You've got a lot of videos to watch, but I want to just touch on the story of how UCNA, a symbiotic cyanobacterium, was discovered. The genetic diversity of this thing called UCNA, the biogeography, nitrogen fixation, and the symbiosis itself, and then a couple of comments about the implications for evolution. So this is kind of a timeline that's published. You can go see it in this Nature Microbiology Review paper, but it sort of shows how this organism that we called UCNA was discovered. And it was discovered by using the polymerase chain reaction to look for a nitrogenase gene. And this little blue set of letters up here is actually the sequence that I amplified with my group back in 1998 that started this whole train of discovery of this unusual symbiosis. So this was started with degenerate PCR, polymerase chain reaction, and we amplified this sequence. And then along the road, we found out that not only was this a cyanobacterium that fixed nitrogen in the oceans, but it was also symbiotic with a unicellular algae in the plankton. And the advent of molecular biology techniques such as quantitative PCR allowed us to look for this organism around the world, many groups, not just us. And the map of the distribution of nitrogen fixing organisms sort of broadened even up into high latitude waters, such as the Arctic Ocean with Shiozaki and Katie Harding, my student's discovery of nitrogen fixing organisms here. And just in a few weeks ago, actually, a paper discovering or reporting the gene sequence for UCNA in the Arctic Ocean, in the Antarctic Ocean as well, around Antarctica, the Southern Ocean. So during most of this time, this organisms continued to be uncultivated, not visible by microscopy. And so we had to use these types of techniques in order to look for UCNA. So how was it done? Well, we didn't have a way to visualize it by microscopy. So we would take seawater samples and run the cells through a flow cytometer and then sort the cells according to their characteristics into different test tubes that we could then test with PCR for looking for the nitrogenase gene fragment. So we could sort these different populations in even finer scale and show where the nitrogen fixing organism was. And so ultimately we were able to show which cells had the nitrogen fixing gene, NIFH. And then we were able to use those sorted cells to obtain the whole genome sequence and a metagenomic analysis of those sorted cells. And then by sorting individual cells from these flow cytometer sorts, we were able to identify a partner that was associated with the nitrogen fixing cell, which is a haptophyte related to coccolithophore. It's called Broadrootosphaera bigelowii. So we use the same PCR technique looking for the nitrogenase gene, but then we also look for the 18S gene of the organism that was associated with that one particle that was sorted by a flow cytometer, and we're able to identify the partner cell. And this is an important paper by your instructor, Rachel Foster. It started a lot of the work on the symbiosis aspects of what we call UCNA. So UCNA, why is it called UCNA? So this organism was a unicellular cyanobacterium that is nitrogen fixing. We studied it by the NIF gene. We called it Group A for a long time, but then Dave Scanlon's group, the Mazard et al. Paper, used 16S primers, and they developed a set of 16S primers for looking for a phylogenetic group, the unicellular cyanobacteria that had nitrogen fixing genes. So they called their primers UCN primers. So I decided it would be better to try to make the nomenclature consistent across different studies. And so this is unicellular cyanobacteria nitrogen fixing Group A, UCNA. And what we found from the genome is that this genome was really unusual. So there are a lot of unicellular cyanobacteria that fix nitrogen. They tend to have larger genomes than, say, Synechococcus and Prochlorococcus. This one is actually the same size as, or even smaller than some Prochlorococcus genomes, because it's missing so many metabolic pathways. And so all these red arrows are things that are missing, and it's missing some very fundamental things like the entire TCA cycle, rubisco, so it can't fix carbon like other cyanobacteria, and photosystem 2 of the photosynthetic apparatus, which means that it can't evolve oxygen photosynthetically, and it can't perform oxygenic photosynthesis. So that told us that it was likely symbiont, and we went to the single cell approach and discovered that the cyanobacterium was actually associated with this haptophyte called Braurutosphaera bigelowii. So two things were happening. People were studying this thing called Braurutosphaera bigelowii, which is an interesting calcifying coccolithophore organism that forms these really fantastic pentalith plates, because it's an important paleo marker in sediments. And at the same time, we were finding these sequences. We got the genome sequence, found out it's a really small genome, that it couldn't fix carbon. And then we found out by doing the single cell technique that it was a symbiont of this haptophyte, and the haptophyte sequence was similar to this sequence that Kyoko Hagino had been studying, and others. And so it was about the same period of time that these two discoveries were being made, and Kyoko was able to use electron microscopy and identify a spheroid body and amplify a sequence that suggested that the symbiosis was in her samples from the coastal waters of Japan, as well as our open ocean waters from Station Aloha. Once this genome was sequenced, then the 16S sequence was available, so fluorescent in situ hybridization could be used, and fish studies allowed the organism to be identified not only by PCR, but by visualization techniques using fluorescent in situ hybridization. So the symbiosis is based on metabolite exchange, first shown by Ann Thompson and Rachel Foster with nanosems and N15 uptake experiments that showed that the cyanobacterium and the haptophyte rapidly became enriched with nitrogen fixed by the cyanobacterium, and the carbon was fixed by the haptophyte, and so they exchanged nitrogen for carbon. But it leaves a lot of complex questions which we're still trying to work on, which is, well, what are the carbon compounds that go from the algae to the cyanobacterium, and what are the nitrogen compounds? Is it simply ammonium from nitrogenase, or is the nitrogen metabolized to an amino acid then exchanged? And we also didn't really know what the physical association was. In the calcified form, it's fairly clear that the cyanobacterium has to be inside the cell. But in the cells that we saw in the open ocean, where we hadn't yet seen any plates, we didn't really know whether it was inside the cell or attached to the cell. This is just showing that there's a relationship between nitrogen fixation and carbon fixation across the different groups of eucin A, which I'll talk about in a minute. So the fish imaging was really important because it allowed us to actually, various people, and Anna Cabello started this along with Francisco Corneo Castillo, who showed that they could visualize cells from samples that were collected in the Global Terra expedition. And what we see is that there's actually a range of size of this symbiosis, with the red being the cyanobacterium and the green being the eukaryote, being identified by the ribosomal RNA genes. And Hagino, again with electron microscopy, is able to show that there are these spheroid bodies which are likely the same cyanobacterium that are identified in fish in plankton samples. So haptophytes have complex life cycles, and with Braurutosphaera bigelowii, this is not completely published yet, but it's believed to be actually have a complex life cycle. It's known coccolithophores that they go through a meiotic life cycle that have somewhat different morphologies. And at B. bigelowii, it's believed to go from a flagellated form to the calcified form. And this is an organism that was identified in the literature as Chrysochromulina, and we now believe that that is actually just one life stage of Brarudosphera bigelowii. And this is all work of Kyoko Hagino. So there are multiple different variations within the UCNA. There are clusters of sequences of slightly different genetic identity, and these group into some different groups that we call UCNA 1, 2, 3, 4. The most common ones being UCNA 1, which we first identified in the open ocean as probably an open ocean form. UCNA 2, which is identical to what Kyoko Hagino was studying in Japanese waters, and is a bigger form, and UCNA 3, which is a somewhat intermediate form that we think is also probably open ocean. And there's sequences that hit at a group 4, and there may be more. Most of these are uncultivated, although as we'll see, there's an exception with UCNA 2. So these different forms, UCNA 1, UCNA 2, are very similar in that they're missing the same things like rubisco and photosystem 2, but they're divergent in DNA sequences. So this is cooler colors are less similar sequences, and so these sequences between comparing A1 to A2 are only 70 or 80% identical, with spots that go up to 90%, 99%. So we think that these were evolved early on, 100 million years ago is the estimate, 112 million years ago. But then that they have diverged and accumulated mutations in the sequences, but have not changed much in the genome content in terms of genes. And we know that these things are widely distributed around the oceans. And the remarkable thing, or the really intriguing thing about UCNA, is it seems to be found in habitats that we didn't normally associate or think about with nitrogen fixation early on, such as coastal regions, high latitude waters found in Danish Strait, Lasse Riemanns group, Arctic Ocean, as I said, and now even near Antarctica, off the coast of Antarctica. So it's very widely distributed and in different habitats. And with nanosems, we can show that it fixes nitrogen. And one of the things it does is it fixes nitrogen only during the day. It doesn't fix at night. And so these are nanosems images showing that the cells get hot or labeled with N15 during the day, but they don't fix any nitrogen at night. And so these are unicellular cyanobacteria. Most unicellular cyanobacteria, since they're individual cells, would fix nitrogen at night to avoid the oxygen they're evolving during the day. But this cyanobacterium doesn't evolve oxygen, and so it fixes nitrogen just during the day. So it's like some of the other nitrogen-fixing organisms, such as the diatoms in the plankton of the oceans that we started off talking about, which evolve oxygen during the day, but they have specialized cells. Trichodesmium fixes nitrogen during the day, but we don't really understand completely how it does so. Crocosphere fixes nitrogen at night. UCNA fixes nitrogen during the day. And although it doesn't evolve oxygen itself, it's living in or very close to a unicellular algae. So it is exposed to oxygen during the day, and somehow it has to avoid oxygen inhibition. How does that work? Well, we're doing some modeling with Mick Follows on how the relationship between growth of the two organisms work, how that controls their relative size. And one of the things that's involved with that is determining how the oxygen dynamics are controlled, because you can't have too much oxygen for nitrogen fixation, but you have to have oxygen to have respiration to have nitrogen fixation. So in looking at this and these different strategies for how to avoid oxygen, Fran was a postdoc in my lab at the time, started thinking about what was the composition of the cell wall of UCNA that might be involved in protecting it from oxygen. And in the literature, he found this terrestrial bacterium, not a cyanobacterium, that uses lipids called hopanoids that slow the oxygen diffusion across membranes. And so these hopanoids have genes that encode those hopanoids, and he looked for the pattern of distribution of hopanoids, and what he found was is that eusin A contains the genes for these hopanoids. So not only does eusin A contain these hopanoid genes, but the other nitrogen fixing organisms that have to avoid oxygen also have hopanoids. And even organisms that just have to organisms such as Trichodesmia and Cyanotheke, they fix at night, but they still have to control the oxygen flow. So what he found was that the pattern of hopanoid distribution suggested that it is involved in nitrogen fixing organisms. You can see that Prochlorococcus doesn't have these hopanoids. It doesn't have the need to protect nitrogenase. So it suggests that hopanoids may somehow be involved in nitrogen fixation protection in eusin A to control the diffusion rate of oxygen. So we're trying to figure out really what is the relationship between these organisms, and we've been working with some collaborators, Kostas Marinas and Himadri Pakrasi, and led by student Debalina Sarkar, who's been using flux balance analysis to really try to understand, well, what is it that it's exchanged? So we know from the genome of eusin A that it's missing a lot of metabolic pathways. And so from their analysis, they know that at least 28 different compounds have to be. So it's not as simple as just giving eusin A sugar. You've got to give them 28 different molecules. So there's got to be a lot of exchange going on across the cell. It's got to be a very complex interaction where many of these intermediates, which include 10 different amino acids, adenine, guanine, and some vitamins, all have to be exchanged. So the symbiosis is a lot more complicated than we thought. It's not just, oh, I fix nitrogen, you fix carbon, and we're happy together. There's an actual very complex network. And that may make sense if you think about it, because the symbiosis can't be lost between the two organisms, or for the cyanobacterium for sure can't live by itself. So it can't be lost just because the nitrogen fixing conditions are not present at any given moment. And one of the interesting things that's hinted at by looking at the metabolic capabilities is that eusin A cannot synthesize a number of vitamins. And, you know, it's given all of the things that it's lost, that's maybe not that surprising. But one of the vitamins it can make is B12. Now, it makes a cyanobacterial form of that called pseudocobalamin that most eukaryotes can't use unless they convert it. But they have the genes for B12. And it's not just one gene, it's a whole set of genes. So given all of the genes that are lost, why would it maintain those? And it turns out that most of the enzymes that require B12 that other organisms have, eusin A doesn't have. So why would it maintain this pathway if it doesn't actually need B12? So this suggests that perhaps the B12 is being made for the host, although the host would then have to convert it. It's going to be a more complicated story, but it again suggests that the interaction between the symbiotic organisms is a lot more complicated than we thought. So here's the exciting news. It's been a couple of years now, but Kyoko Hagino has been maintaining a culture of eusin A2. This is the coastal form, and so it's a little bit larger. But you can see that it's this this stage of it is not always calcified. It's in this free-living flagellated form, like a typical haptophyte. And here you can see the cyanobacterium, just like an organelle inside the cyanobacteria. So it's a very exciting time, because although this is not the oceanic form, which is really only about this big, it does give us the possibility to do structural analysis, biochemical experiments, isotope experiments. Very exciting, and hopefully this will help us learn more about how the symbiosis works. So finally, I just want to say that the real question here is, what is this thing? Is it something that just is a symbiont, presumably an obligate symbiont, an endosymbiont, or is it actually an organelle or approaching an organelle? And the analog is a non-nitrogen fixing cyanobacteria that is associated with a protist called Pollinella. So this is, or was, a cyanobacteria. It now too has gone through genome reduction, and it's at the edge, and people argue whether it's either an endosymbiont or an organelle. And it has characteristics such as there has been some exchange of genes between the genomes of the host and the cyanobacteria, and there are mechanisms for transporting proteins from the host into the cyanobacterium, all of which are hallmarks of a very tight relationship, such as you would find in an organelle. So the question is USIN A, where is it along the road from being an endosymbiont to an organelle? But what we do know is it's widely distributed and an important contributor to the global marine cycle, and that the symbiosis is maintained by multiple mechanisms. It's not a simple nitrogen versus I give you nitrogen, you give me carbon. Is it an organelle, or how far along the road is it? And we're looking for gene transfers and things like that. And I guess I'd like to say that this story suggests that there's things out there yet to be discovered. I mean, we sort of initially only found this because we were looking for a gene. If we would have been looking through the microscope, it would have been a long time. Now this thing is showing up, the USIN A is showing up in metagenomic surveys and so on, but it wouldn't have been known what it was if we hadn't done some of the early studies where we knew what we were looking for. So it's a very exciting time. There's a lot of things to do. And I hope that some of you end up being those who discover things. There's been many different people and funders along the way, too numerous to mention. I can't name names because there'd be somebody who's missed current lab members, but Dave Carl was very supportive of our early surveys at Station Aloha, continues to be a collaborator. And I just want to put in one plug here for a book that should be published by the end of the year on marine nitrogen fixation by Doug Capone and I. So thanks for your time. Sorry it went a little long, and I hope you got something out of it. And I will be seeing you in our discussion section.